Hello, everybody. My name is Anne Levy. I am the Senior Program Manager for the Center for Faculty Development, and in that role, much involved in the Anne Klibanski Lecture Series. In today's session, Dr. Sian Piret from Stony Brook University is going to present on the role of branched chain amino acid catabolism in healthy and injured kidney. Dr. Piret has disclosed that she does have one financial relationship, but it does not conflict with today's lecture. Before I hand the virtual microphone to Dr. Piret, I'm just going to share a very brief history of the Anne Klibanski Visiting Scholar Award Program. It was created by our former CFD director, Miriam Bradella, to offer MGH women faculty the opportunity to serve as a virtual visiting professor and give grand rounds or other special lectures at a national or international institution organized by that scholar's champion, a senior faculty member at MGH, with the help of the CFD. This program and accompanying lecture series is named after Dr. Anne Klibanski, who is the current president and CEO of Mass General Brigham and former director of the Center for Faculty Development. A highly successful researcher, Dr. Klibanski has been a strong supporter and advocate for women. We've had 4 cohorts of Anne Klibanski Scholars to date. We'll show you all of them. The inaugural one from 2020 to 2021 had a cohort of 36 scholars. The second one, 21 to 22, those years, had 32 scholars. In 2022, 2023, we had 31 scholars. And finally this year, but it's not final, we will continue. Our 4th year, we selected 29 scholars. Dr. Kamalika Mukherjee from this year's cohort will be introducing today's speaker from Stony Brook, the institution that hosted Dr. Mukherjee's virtual talk as part of the Anne Klibanski Visiting Scholars Award Program. This program would not be successful without the commitment and energy of the faculty we call our Anne Klibanski Scholar Champions, who help the scholars find exciting speaking engagements. Now, the goal of the Anne Klibanski Visiting Lecture Series is to catalyze and foster collaborations across national and international institutions. We have a slide that shows a few of the institutions we've collaborated with over the years. Highlighted in yellow is Stony Brook University to represent Dr. Piret. Now I'm going to pass the microphone to Dr. Kamalika Mukherjee from NGH to introduce you to today's speaker.
Thank you, Dr. Levy. It is my absolute pleasure to welcome Dr. Sian Piret. Dr. Sian Piret is an assistant professor of medicine in the Division of Nephrology and Hypertension at Stony Brook University. She earned both her Master of Biochemistry degree and PhD from the University of Oxford. Following her doctoral studies, she conducted postdoctoral research at the Academic Endocrine Unit, Oxford Center for Diabetes, Endocrinology, and Metabolism. In 2017, Dr. Piret joined Stony Brook University as a postdoctoral research scientist and research assistant professor. She then became an assistant professor in 2020. Her research focuses on understanding mechanisms related to both acute kidney injury and chronic kidney diseases. Dr. Piret has received notable recognitions, including the NIDDK's R01 grant, AHA Career Development Award, and the Joseph V. Bonventure Research Scholar Award from the ASM. Without further ado, I would like to extend an invitation to Professor Piret to share her research with us.
Well, thank you so much for the kind introductions. It's really a pleasure to be here. Thanks to Dr. Levy for the invitation. And we really enjoyed hosting Dr. Mukherjee back in December when she sort of visited us virtually to give her talk. So it's really a pleasure to be able to return the favor today and be sort of visiting with you all. So I'd like to tell you about some of our studies on, as Dr. Levy mentioned, the role of branched chain amino acid catabolism in healthy and injured kidney. And I'll say up front, when I was an undergraduate doing biochemistry, I hated cell metabolism. So the fact that I now spend my time studying it is kind of ironic. And I'm sure my undergraduate tutors would laugh at me for trying to do it. So here goes. Just for any who are not nephrologists in the audience, acute kidney injury is really defined as a sudden reduction in kidney function, and it's usually measured by increased plasma creatinine or decreased urine output. And broadly, it can be defined into three kind of broad causes: pre-renal causes such as severe hypovolemia, for example, intrinsic causes, which are obviously intrinsic to the kidney itself, which is what we mainly focus on. This can be ischemia, sepsis, exogenous or endogenous toxins, and potentially post-renal causes such as obstructions. And really, there are a number of possible trajectories that can follow on from an episode of AKI in different patients, depending on both severity of the injury, but of course on a lot of intrinsic factors that we don't yet understand. So in some circumstances, for example, this purple line, even a relatively severe hit of AKI can actually resolve quite quickly and to all intents and purposes leave no residual loss of kidney function. But kind of at the other extreme, a severe hit of AKI may actually never resolve and may always subsequently result in kidney dysfunction. And there's any number of different trajectories between those two kind of extreme outliers. But whatever happens, even a single episode of AKI can substantially increase the risk of developing CKD, chronic kidney disease, or even end-stage kidney disease subsequently. So this is probably anybody who's a nephrologist in the audience has probably seen this figure multiple times, but this is a meta-analysis that was done several years ago. The top panel shows the excess risk of developing CKD after AKI, and the bottom shows the risk of developing end-stage kidney disease after a single or multiple hits of AKI. So it's really quite a substantial effect size. And you know, the patient factors that define the way that a patient's trajectory is going to follow after AKI are really not well defined at all. And of course, if we could define them, we'd probably be able to do something about it. The same is true on a cellular level as well. So the proximal tubule, which is the part that I work on, is really particularly susceptible to AKI. And these cells really undergo a whole host of changes in response to an acute kidney injury, some of which are depicted here. So, for example, proximal tubule cells are normally tall columnar epithelial cells with a brush border on the apical side and a very well-defined polarity. During AKI, this polarity sort of disappears. The cells de-differentiate. They lose their polarity such that some basolateral membrane proteins become missorted to the apical membrane. Some apical membrane proteins become internalized into the cell and sequestered there. In some circumstances, these cells can actually re-enter the cell cycle as well. So proximal tubule cells normally post-mitotic, non-proliferating, terminally differentiated. But in injury, there's evidence that some of these can actually re-enter into the cell cycle. And the idea is that cells can do this to divide to replace cells that have been cleared either through apoptosis, necrosis, and therefore repair the injury. However, a subset of these can actually get stuck in a G22M stage at rest. And this is really the cell type that's thought to be associated with secretion of pro-fibrotic and pro-inflammatory factors that can potentially define the transition to fibrosis after an acute kidney injury. And then the part that I'm really spending most of my time on is the actual cellular metabolism by which proximal tubule cells generate ATP. So obviously proximal tubule cells have a very critical role in bulk reabsorption of solutes, and this is really dependent on the sodium-potassium ATPase at the basolateral membrane. They therefore have to generate large amounts of ATP to be able to achieve this. So usually proximal tubule cells utilize fatty acid oxidation through mitochondrial beta oxidation, feeding into the TCA cycle, and there through oxidative phosphorylation to generate ATP. However, in the setting of injury, this whole process of fatty acid oxidation is really strongly suppressed, both through direct mitochondrial damage, which happens during injury, but also through downregulation of critical genes encoding critical enzymes in those pathways. So instead, fatty acids accumulate in the cell. We can see this happening in lipid droplets. And the thought in the field has been for quite some time that maybe proximal tubule cells could kind of switch over to glucose metabolism, so glycolysis instead, a kind of Warburg effect type scenario, which would obviously generate lactate as an end product. I'll show in a minute. This is a little bit equivocal. This is not sort of a kind of fixed thing. There's some evidence to suggest that that's not necessarily the case. But again, similarly to the whole patient level, what defines whether these cells can repair or whether they undergo this sort of unsuccessful repair state leading to fibrosis, the cellular factors that influence this also are not well understood. And again, this is obviously thinking about development of therapeutics. This is where we might be able to intervene to promote a successful repair state rather than an unsuccessful repair and fibrosis state. So we've been looking at a number of kind of key questions in this area, which I'll tell you a little bit about each of these today. All of these questions are still unresolved. We're still working actively on all of them. None of them is a complete picture yet, but I want to sort of give you an idea of where we are. So as I mentioned, fatty acid oxidation is strongly downregulated. So key question is, what are the potential other metabolic pathways that are active in the kidney, and particularly the proximal tubule, which might be altered in injury and disease that we also might be able to manipulate? How are these pathways regulated, both in healthy kidney and in injury? What are the downstream consequences of altered metabolism? So as we'll see, metabolic intermediates and metabolic pathways can have a number of other effects on other cellular processes, not just about making ATP. And of course, the million-dollar question, can manipulation of these metabolic pathways alter outcomes after injury? So I'll walk through each of these in turn. So what about other ways in which proximal tubule cells might be able to generate ATP? Well, I've kind of hinted about this already. Normally in proximal tubule cells, fatty acid oxidation predominates, generating acetyl-CoA, feeding into the TCA cycle and generating ATP. Glycolysis is actually, not only is it absent, it actually goes in the reverse direction. So proximal tubule cells actually do gluconeogenesis to generate glucose. During injury, fatty acid oxidation is severely downregulated, and it's possible that glycolysis could be upregulated to compensate. And then we've got this other sort of very broad and hand-wavy category, catabolism of amino acids. So it's known that, for example, glutamine can be used by proximal tubule cells to generate glucose. So there are other effects going on there. So what about glycolysis? Well, for a long time, people thought that this was upregulated in injury because if you look at expression of key glycolytic enzymes in the setting of injury, their expression levels go up. So this is a mouse model of sepsis, CLP, versus sham mice. And these are some key glycolytic enzymes. So you can see that both at the mRNA level and also at the protein level, these enzymes are strongly induced. So it would make sense that there would be more flux through this pathway. However, when you look at the actual flux, that doesn't necessarily turn out to be the case. So these are measurements of extracellular acidification using a live cell metabolic flux analyzer, a seahorse. And basically, if you see more acidification, it implies that there's more glycolysis, which generates lactate, which of course is an acid. And in fact, you can see that in the sham animals, compared to the sepsis mice, the CLP, actually in the CLP mice, it's actually lower. It's not being induced. If anything, it's actually suppressed as well. So this is kind of thrown into some doubt, actually, how much glycolysis is really going on. And this was in isolated proximal tubules from normal or septic mice. So we got interested in a particular subset of amino acids called BCAAs, the branch chain amino acids. These are valine, leucine, and isoleucine. They're essential amino acids. You can't make them. They have to come from the diet. And right when we were actually starting to get interested in this very helpful paper came out showing that the kidneys indeed do use BCAAs as an energy source, which hadn't really been shown before. So just to remind you, this is the branch chain amino acids, leucine, isoleucine, and valine. And what this study did was they don't work on kidney, they just work on BCAAs. They took mice, they took heavy carbon-13 labeled BCAAs, a mixture of leucine, isoleucine, and valine, and they just infused them into mice and collected a whole bunch of organs and basically had a look to see where is that carbon-13 label going. Is it going into protein synthesis, which of course is the other place amino acids can go, or is it going into TCA cycle? And then they were able to calculate this oxidation flux. So in other words, looking at the amount of BCAAs that showed up in the TCA cycle intermediates and looking at the relative organ masses and figuring out a kind of flux measurement. And in fact, if you look at the numbers that they came up with, it turns out that the kidneys use quite a lot of these things. So it's actually only about a third behind the heart, the brown adipose tissue, and the pancreas, that the kidneys are actually utilizing quite a large percentage of these BCAAs specifically to power the TCA cycle, which implies obviously that you would generate more ATP from them. So this was great for us because we were just getting interested at the time. And the reason that we were getting interested in them is because we found that a lot of the key enzymes in this pathway are actually downregulated in the setting of at least of mouse AKI and fibrosis. So here's a kind of schematic of the BCAA catabolic pathway. So we're going from branched chain amino acids right at the top here, all the way down to acetyl-CoA and succinyl-CoA, which of course feeds directly into TCA cycle and even into oxidative phosphorylation. And there's a load of enzymes here, which I haven't bothered labeling more because it gets too complicated. The key step right here, and this is a mitochondrial pathway, I should say. Key step right here is this BCKDH complex, which I'll talk a bit more about later on, which is this large multi-subunit complex. This is the first irreversible step in the pathway. So the very first enzyme is freely reversible. The second enzyme, this BCKDH complex, is the first irreversible step. So once BCAAs have gone through this step, they're committed to the rest of the pathway. They can't go back. And not only that, it's actually the rate limiting step as well. So the flux through the whole pathway, to a large extent, is defined by the flux through this particular enzyme complex. So what we found was that in various different types of injury in mice, a number of these amino acid the genes encoding these amino acid enzymes are severely downregulated. So here are a number of the enzymes which are labeled on the pathway. AA1 is a proximal tubule toxin called aristolochic acid 1, which I'll describe more quite soon. We did an injection followed by 24 hours and 48 hours, and then a series of 2 and 8 injections. And you can see even after 24 to 48 hours, some of these enzymes are downregulated, which actually precedes proximal tubule cell death. Cisplatin, obviously, we know causes AKI. Again, there's a strong downregulation. I think this was after 8 days of cisplatin. And then unilateral urethral obstruction, so a kind of obstructive model, which is more of a fibrotic model. Again, strong downregulation. And then LPS, which is a sepsis model, same thing again. And this was just whole cortex at the time, but other groups have subsequently shown that this is proximal tubule specific. So this is a paper from Ben Humphrey's group, showing you can see these enzymes here are quite highly expressed in healthy proximal tubule segments all the way through, whereas in all of these various injured types of cells, this expression is strongly suppressed. And as I showed you before with glycolysis, just because you have changes in enzyme levels doesn't necessarily mean a change in flux, but there's also some metabolomic studies out there now which are starting to show the same thing in terms of the metabolic intermediates. So we think it's real. And then we actually looked in human AKI and CKD data sets as well, and we actually found the same thing. So this is a data set which is available online, which my postdoc, Sama, has been reanalyzing. If you just take the differentially expressed genes in AKI and just run an unbiased pathway analysis, valine, leucine, and isoleucine always comes up near the top. And in fact, it's even higher than, even more significant than fatty acid degradation. And again, it's a lot of the same kind of enzymes that we see in mice that have that differential expression. And then we reinterrogated a human CKD data set available on NephroSeq. And what we found was actually the expression of these enzymes actually correlated with GFR. So patients with high GFR actually have pretty high expression of all these enzymes. As you go down through decreasing GFR, you can see that the expression of these enzymes gradually is reduced. So it seems to be the case in both mouse and human, and also in various different types of injury as well, and in the setting of acute kidney injury and the setting of chronic kidney disease. So this is really curious, and nobody's really studied it before. I'm sort of going to take a backward step to kind of tell you a bit about how these pathways are regulated, and this is kind of telling you how we got to the BCAAs in the first place. Actually predates the BCAA work that we did. But when I joined the lab here at Stony Brook, the lab that I joined of Sandeep Manipatu was looking at this class of transcription factors called Kruppel-like factors. So these are a family of transcription factors. They have really hugely diverse roles in cellular physiology. Pretty much anything you can think of, there's a KLF in there somewhere. And the interesting thing about these is that they can both transcriptionally activate and repress target genes. So they can work in different ways. And, you know, a particular KLF can both activate and repress the same target gene in different tissues. So it's also very cell specific. Just in case anybody needs a reminder of how these transcription factors work, they bind to a specific sequence in DNA, and along with normally with cofactors, they either will activate genes or they will repress gene expression. And these KLFs are three zinc finger transcription factors. So they have these three very highly structured zinc fingers at the C-terminus, which interdigitate with specific sequences in the DNA. So if you just look on a global scale, transcription factors obviously are master regulators of lots of different cellular processes. So they tend to come up a lot when you look at just wide-scale sequencing, whether that's in mice or humans, whether it's single cell or bulk, whether it's IRI, sepsis, whatever the type of injury is. But these KLF families keep coming up. So for example, here's KLF15, which is always downregulated in injury. And we've done a number of studies showing that KLF15 actually plays a role in fatty acid oxidation in the proximal tubule, which I'm not going to have time to tell you about today. But the other one that always comes up is KLF6. It doesn't matter whether you look at mouse, human, type of injury, acute, chronic, it always comes up and it's always very strongly upregulated. And this is a specifically proximal tubule effect. So before I tell you about our work on KLF6, I just want to kind of go into a bit more detail about this toxin that we use, because not everybody has come across it before. So we use this substance called aristolochic acid. It's actually a naturally occurring toxic compound. It's found in the Aristolochia plant species. And it's actually clinically relevant as well. So you can come across it in two sort of clinical contexts. One is a result of acute ingestion, which accounts for the so-called Chinese herbs nephropathy or AA nephropathy, versus a chronic ingestion, which is responsible for Balkans endemic nephropathy. The Aristolochia plant seeds get into the bread crops and people actually consume it. In the acute stage, it leads to low molecular weight proteinuria, mild hypertension, anemia, and then it can progress to end-stage kidney disease. And the reason that we like it is because we're studying proximal tubule, and this is highly proximal tubule specific. So it actually enters the proximal tubule through these basolateral organic anion transporters, and then it forms these covalently bound DNA adducts, which can actually persist in the cell for quite some time. Some of them get repaired, but a lot of them persist. Also, it causes direct mitochondrial damage, which of course produces reactive oxygen species, which can further damage DNA. It also inhibits the process of receptor-mediated endocytosis, which is how proximal tubule cells reabsorb small proteins which have been filtered by the glomerulus. And then any that's not otherwise bound in the cell gets extruded through the multi-drug resistant proteins into the tubule and then the urine. And the other reason we like it is because it induces a very nice AKI, but also fibrosis in mice. So a lot of the time when you see cisplatin being used in mice, which is also a proximal tubule toxin effectively, you get a really nice AKI, but it doesn't always transition to fibrosis. There are some milder protocols out there now which do this, but aristolochic acid is very, very reliable at doing that. And just to give you an idea of how this looks, we use this in a kind of multi-hit, so several hits of AKI every 3 days apart. And then we can either stop the study, very acute phase or after a series of injections, or we can actually then stop the injections and let the mice sit for whilst the fibrosis develops. So after a single injection, there isn't really a lot of cell death. There's a little bit of loss of the brush border shown here in the dark pink, but it's really very mild. After 2 injections, we start getting quite extensive proximal tubule cell death, as you can see here, starting to get some protein casts forming. And then after a series of multiple injections, what we find is that a lot of the proximal tubule mass has just disappeared. And what you're left with is effectively the denuded basement membranes here, as well as a quite robust inflammatory response and, again, protein casts. Interestingly, glomeruli are basically unaffected. And then if we allow the mice to sit around for a bit, we get quite an extensive fibrosis. We get basement membrane thickening, again, a very robust inflammatory response. So our question was, what is the role of proximal tubule KLF6 in injury? It goes up no matter what type of injury you use. What happens if we take it away? So that's what we did. We generated mice lacking KLF6 in the proximal tubule. And we found that this is really interesting, that KLF6 is actually a detrimental effect in kidney. So when you take it away, it's actually protective. So when you administer AA, obviously you get a rise in serum creatinine and serum urea nitrogen due to the kidney injury. This is attenuated in knockdown mice. Here, what we're doing is staining for the brush border of mature proximal tubules with lotus lectin, and co-staining with an injury marker called cytokeratin 20, which gets switched on de novo in injured proximal tubules. And you can see in the control mice here, you've lost most of the proximal tubule mass, whereas this is significantly preserved in the knockdown mice. There's also a less robust inflammatory infiltration in the knockdown mice. And then at the end of the fibrosis phase, we can see that there's really extensive fibrosis here in blue in the control mice, whereas this is attenuated in the knockdown mice. So KLF6 is really, loss of KLF6 is protective against injury. So it goes up in injury. When you take it away, it's actually beneficial, which is kind of interesting biology anyway. So at the time, we did bulk RNA-seq to try and figure out what's going on, what pathways might KLF6 be regulating. And obviously, since KLF6 is a transcription factor, we can look at where in the genome KLF6 binds. We can cross-match that with the changes in gene expression in control versus knockdown mice. And when we match the two up, we can actually get some idea of which of those genes might be directly regulated by KLF6 versus those that might be indirectly regulated by KLF6. And so this is just a heat map of all the gene expression changes, so wild types and knockdowns uninjured, and then wild types with injury and knockdowns with injury. So you can see here that there are quite substantial gene expression changes. We actually focused our attention down here on genes that were downregulated in injury, but which were less downregulated when we removed KLF6. And when you take all of these genes, 388 genes, you stick them through a pathway analysis, up at the top, we get valine, leucine, and isoleucine degradation, which is what put us onto this in the first place. And in fact, all of the top hits are metabolic pathways. So this is specifically comparing what happens when you take away KLF6. These genes are all becoming switched on again, which suggests when KLF6 is high, it's actually suppressing the expression of all of these genes. And then what we did was we sort of took it a step further and we categorized all of these genes, depending on whether or not they had a KLF6 binding site close to their promoter. In other words, are they likely to be regulated by KLF6 or not? So in this first category here, we had genes where there's at least one KLF6 binding site within 1 kb of the transcription start site. So really, really close to the transcription start site. If a transcription factor binds there, there's a pretty good chance that it's really doing something biologically relevant. And then the second category was a binding site between 1 to 10 kb further away, so a little bit further out, so possibly less confident. And then a whole bunch of genes here which just didn't have a KLF6 binding site at all, so any effects here are likely to be indirect effects. So when we took these genes, specifically this class 2 genes with a KLF6 binding site really, really close to the start site, we only had 103 genes. And yet, we actually retained the significance of some of these pathways. So, and particularly the valine, leucine, isoleucine degradation, and some other actually amino acid pathways. So what that implies is that even though we've now only selected a subset of 103 genes, in that 103 genes, we've actually enriched for genes that are in this pathway compared to the original 388 genes. So it suggests that KLF6 may have a role in regulating this pathway. And just because, for my mind, it's easier to look at an actual kind of pathway map rather than just a list of gene names, we actually plotted these onto some metabolic maps. So what you're looking at here is the comparison between injured and uninjured control mice. And the arrow colors denote the RNA-seq fold change. So green here is downregulated, red would be upregulated. And then the box color for each enzyme denotes, again, those KLF6 binding site classes. So the purple ones have a KLF6 binding site very, very close to the start site. Blue is more distal, white is none. And I hope what you can see is that a lot of the downregulated enzymes here with the green arrows also have KLF6 binding sites somewhere close to their transcription start site. So they sort of correlate. And again, here at the bottom, we've got these feeding into the TCA cycle. And then what happens if you compare the knockdown mice to the control mice? A subset of these come back on again. Their expression is actually up in the knockdown compared to the control. And again, these almost entirely correlate with genes with KLF6 in their binding sites and their promoters. So it really looks like it certainly correlates. We can show both in mice and in cells that when we manipulate the KLF6 levels, we also manipulate the expression of a lot of these enzymes. So this is actually after 3 weeks of AA. We've got our knockdown and control mice. You can see in the knockdown mice, expression of several of these genes is somewhat restored. This is all relative to one being uninjured. So they're still down, but they're at least restored a little bit. This is in HK2 cells expressing control vector or overexpressing KLF6 with AA. And again, you can see that there's a further suppression when we overexpress KLF6. So what this is really telling us is that KLF6 seems to suppress the expression of these enzymes. And then we did some in silico work looking at, again, the locations of KLF6 binding sites. And we can see that there is one right here in this key enzyme BCKDHB, and it falls within a region of open chromatin. So we think that KLF6 binds there. We confirmed this by CHIP, qPCR, so chromatin immunoprecipitation. So when we treat with AA, we get an increased binding of KLF6 to this area. And when we further overexpress KLF6, we get a further binding of KLF6 to this region. So we're actually testing the binding right here. So we think KLF6 is actually regulating these pathways directly. We're trying to show this now a bit more directly through luciferase assays. So again, when we overexpress KLF6, we would expect the luciferase activity to go down. And then we're thinking about looking at other genes in that pathway as well. And then we actually want to look at some actual flux through the pathway in control cells and cells expressing KLF6. Okay, so I've told you that BCAA catabolism is downregulated in injury, and that we think that this is regulated by KLF6 expression. But what about the downstream consequences? Does it really matter if you lose BCAA catabolism, and if so, how? So obviously I've told you about one of them, which is that we've now lost another potential source of ATP production. And that could very well be important. But our other hypothesis is almost like the flip side of that. So if you have a certain amount of amino acids in the cell, which are normally broken down into other metabolites, if you suddenly put a block in that pathway, you are then going to get probably accumulation of those whole amino acids that are no longer being broken down. And so could it be that actually those whole BCAA, that valine, leucine, and isoleucine, could actually have a toxic effect on the cell? So we've got a loss on one side, but the flip side of the coin is that could we actually have a toxic sort of gain of function, if you like? And there are lots of different things that could be the case, but one that we're focusing on is actually activation of mTORC1 signaling. So it's known that leucine is a very potent activator of mTORC1 signaling. And we know that mTORC1 signaling is also active in the setting of AKI. It's been shown many times. And that this likely has detrimental downstream effects. And so we're interested in whether the accumulation of leucine may actually be one of the things which is promoting mTORC1 activity in the setting of AKI. So just to quickly touch on the first one, do we reduce ATP production when we lose this pathway? We generated cells with knockdown of this key enzyme subunit here, BCKDHB. And then when we use the flux analyzer, the seahorse, to look at ATP production, we find that indeed, when we knock down BCKDHB, we do get a significant reduction in ATP production by these cells. Interestingly, when we did this in the setting of AA, we didn't get a further suppression. So the suppression that we had with AA actually was mimicked by just knocking down that particular enzyme. So we think it does have an effect on ATP production. But what about mTORC1 signaling? So just to kind of very briefly walk you through how this works. Normally, if you are in the setting of low leucine and other factors such as growth factors, mTORC1 would be in an inactive conformation. The reason is because this RAG-AB complex is in its inactive conformation because it's held there by its inhibitor GATA1. The reason that GATA1 is free to do that is because its inhibitor GATA2 is sequestered away by the cestrin-2 protein. In the presence of leucine, leucine actually binds to cestrin-2, relieves the repression of GATA2, which leaves it free to repress GATA1. That relieves the GATA1 repression on RAG-AB, and it allows mTORC1 to become active. when mTORC1 is active, it gets recruited to the lysosomal membrane. So if you see mTOR on the lysosomal membrane, you basically know it's active. So we've done some work in some primary PT cells with AA, and we can show that this actually happens. So in control cells, LAMP2 is the lysosome marker, and obviously mTOR, there's no particular correlation. They're just kind of all over the place. As soon as we start treating with AA, we get these very large lysosomal bodies, which also have mTOR co-localized with them. And then we can also show that there's activation of the downstream signaling marker, which is phosphorylation of S6. That becomes active in the setting of injury. And then we've also been able to show that this happens in proximal tubules in mice. So here we're staining for lotus lectin at the brush border. mTOR again is in red, and LAMP2 this time is in cyan. And what we can show again are these very large lysosomal inclusion bodies, and right on the membrane is mTOR. So we think this is active in our mice in the setting of AA as well. So what we're working on at the minute is trying to make that connection between the increased leucine levels and the increased mTOR signaling. So for example, if we remove leucine and we treat with AA, do we then abrogate the activation of mTOR? And we can also interfere with leucine sensing as well by cestrin-2 to see if that interrupts that connection. And then, of course, we're going to want to know what pathways are downstream of mTORC1 signaling that we might want to manipulate therapeutically. So just in the last few minutes, I kind of want to come to, in a sense, the really key question. These metabolic pathways, fine. Is it just a correlation? Is it actually promoting AKI? One way of addressing that question is, if we manipulate them, do we then alter outcomes after injury? And of course, that leads on to potential clinical applications as well. So to kind of draw everything together, normal kidney does utilize branched chain amino acids for ATP production. And during both AKI and CKD, BCAA catabolic genes are downregulated, or genes encoding those enzymes are downregulated in both mice and humans. And we think this is downstream of increased KLF6 expression, having a suppressive effect on expression of those enzymes. We think downregulation of BCAA catabolism does decrease ATP production. And we think it probably causes accumulation of BCAA, which might actually activate mTORC1 signaling. And our preliminary data shows that activation of BCAA catabolism actually can protect against AKI in mice. So to kind of pull all these together and sort of show you how they fit, AKI suppresses BCAA catabolism, probably due to induction of KLF6. We're looking at both sides of the coin in a sense of cellular mechanisms, in the sense of decreased ATP production and increased mTORC1 signaling. We're also kind of looking at the whole body physiology by hopefully studying these new BCKDHB proximal tubule specific knockdown mouse model to see if there's really a causal link with AKI. And then the sort of therapeutic side of things, we're looking at BT2 treatment, which we think seems to mitigate AKI. We're not sure if that's through ATP production or mTORC1 signaling at the minute. That's something we're trying to tease out. The other thing we're trying to do is actually, it turns out BT2 is not a very good drug. It's not very soluble. It's not very bioavailable. It has lousy pharmacokinetics. So one thing that we want to do is actually derivatize BT2, because we know what the structure is, we know where it binds, to try and generate some improved derivatives. In order to do that, we have to actually design a high throughput kinase assay, first of all, for screening potential derivatives before we go ahead and test them all in cells and mice. Yeah, so just to acknowledge the folks who are wonderful enough to put up with me. My current group is Sama, who's been working on the BT2 stuff, and Louis, who just joined the group recently, is going to be working on the luciferase assays and looking at KLF6 regulation in this pathway. Manasa, who just finished her degree in December, who's coming back as a technician, she's working on a slightly separate project, which I haven't had a chance to tell you about today. And Asher is an undergraduate in the lab, and my mentor here at Stony Brook is Dr. Sandeep Mahapati, who some of you might know. Our collaborators, and then my current and previous funding. Thank you so much for your attention. I'll be more than happy to take any questions you may have.
Thank you, Sean, for the wonderful talk. I don't see any questions in the chat, but if anyone has any questions, please go ahead.
So, hi, it's Lorraine Schratz. I'm a pediatric cardiologist here at MGH, and I also do preventive cardiology. And so I'm not really sure if I have a question because your work is so elegant and way above my understanding and knowledge level. I would just make the comment that elevated branch chain amino acids are elevated in obesity and in cardiac disease. And so your work just makes my head start spinning with thinking about like, how is this all related? So you can comment on that because you probably know way more.
No, thank you for that comment. That's really actually perceptive, because a lot of the work that's come about so far in terms of BCAA catabolism and alterations actually comes from the cardiac field. So it turns out that in, for example, in cardiac ischemia, BCAA catabolism goes down and accumulation of them has been shown to be detrimental and all sorts of things. So actually a lot of what we're kind of writing from the back of is coming from, you know, the cardiac myocytes and that kind of thing. So you're absolutely spot on. In terms of obesity and circulating levels, one thing we get asked a lot is about dietary restriction of BCAAs and whether that would have a beneficial effect. And I don't have a good answer for that part. I think it depends what we find in the sense of cellular mechanisms. If the main issue is loss of ATP production, then restricting those BCAAs is probably not a good idea. If the main issue is actually the accumulation of the BCAAs causing other downstream effects, then yes, dietary restriction probably would be a good idea. So I think from the point of view of obesity and circulating, and you know, if you do a search, Google search online for BCAA, you come up with the first like 100 hits are supplements that you buy off the shelf and people take, you know, and I'm thinking, my goodness, I'm not sure if this is such a good idea. So we just don't know yet, I think. Yeah, obesity, circulating levels. We don't think the kidney contributes to circulating levels as far as we know. That's probably more liver and skeletal muscle. But yes, it's something that is obviously a very fruitful area to explore in the future.
Thank you for the comments. Again, it's like far beyond my knowledge base, but I appreciate the work you're doing. It's really quite elegant, and I appreciate you coming to talk to us.
Thank you. It's very kind of you.
I'm going to second that emotion above me, but I work on Huntington's disease, which may seem really far out of the loop here. But one of the interesting things, I do a lot of omics data, and I've gotten very interested in the metabolism, which, you know, Huntington's disease is a metabolic disease, really. And one of the number one pathways that keeps coming up is branch chain amino acid metabolism, okay, which I know nothing about. And I can tell you hardly anybody in our field does either. So I was just trying to learn. And this has been really fantastic. You're a beautiful speaker. It was a great presentation, even for someone who knows very little about it. So thank you. Thank you very much.
Well, thank you for saying that. And that's really, you know, the more we look at these things, I didn't know anything about them either. So I'm in exactly your position as well. I'm learning on the job, honestly. So, you know, and nobody had looked at them in the kidney before. So we're pretty much the only people doing it. So in that sense, it's exciting because there's a lot to explore. But in the other sense, there's not a lot to kind of grab a hold of. So I absolutely hear you. And you know, if we can be of help in any way, then please obviously reach out, and that would be fascinating. Yeah,
I'm going to take you up on that, and I'm gonna follow up with you. I have to say that the interesting thing is we also have a lot of transcriptomic data that we can mine from various sources. And the first thing I'm going to go do is look at the cripple factor-like proteins and see if they're changed at all. So I mean, there's really a lot to think about here.
Yeah. They also pop up all over the place. I mean, KLF4 is one of the Yamanaka factors that you put into cells and make them iPS cells. So, I mean, they pop up all over the place. I couldn't tell you about KLF6 in the brain because I don't think I've ever looked. But yeah, one of them, there's 15 of them. So you're likely to find one somewhere, or 16. So you're likely to find one somewhere along the line. Yeah. But yeah, and you know, we've also done the same thing. We've gone and mined other data sets and found that BCAA catabolism keeps popping up in human, in mice, and different injuries, and at least in the kidney anyway. So it seems like they're kind of understudied, but I think it's, yeah, again, probably a fruitful area if you want to get a handle on it.
Well, there's definitely been a recent understanding that there's a change in fatty acid beta oxidation. Yeah. So that's I'm seeing these links come. That's what I have been looking at, but I always knew this fatty acid metabolism was changed. And I was like, I don't even know where to begin. There's the metabolism is a whole nother thing. So this like really brought the intersection of both those things. So it was really very timely for me. Thank you.
If there are no other questions from the audience, I just have a comment. Exceptional talk, of course, and the question I wanted to ask, you kind of addressed it in your future work. It'd be really interesting to see if we were to treat these mice who were already treated with aristoclic acid, how would they respond once we, treated them with BT2 or a drug, as you say, that's more soluble, more bioavailable? Those would be interesting data to kind of look into. But yeah, it's exciting and awesome, and we should talk about certain other. Awesome. Thank you so much, Sean.
Sure. No, and you're right about the, treating after the injury. I mean, most of the time we don't know that someone's going to get an AKI, except in very specific cases. But, so that's really our next step is if we give the injury first and then intervene later on, does it still work? And that's kind of a key question, definitely.
Dr. Piret, this has been exceptional. We've had a small audience, but I can tell that everybody who did come was really excited by what you talked about. And what I love about these lectures, your lecture just brings out a shining example of this ability and possibility to connect. Everyone is so specialized that they get stuck in their little cubby, but the body doesn't work like a cubby. So your work is inspiring, and I'm so excited to see that it's already affecting how people think and what they may look into now. And I hope it builds connections and collaborations. I know that you're already going to collaborate with Kamalika Mukherjee. So thank you so much for coming. We deeply appreciate it.
It's very kind of you. Thank you. And thank you so much for the invitation. I really enjoyed talking with you all. So.
Of course. Well, I wish everybody here a wonderful rest of your day. And please fill out the form you're going to get to evaluate this event. Thank you, Kamalika, for being here to introduce Dr. Piret. Goodbye, everybody.
Pleasure.
Thanks.
Thank
Have a
you.
great afternoon.
Bye.